All right. So we know that we're increasing the transcription of this alternative promoter of PGC1 alpha. And so the question is, what's involved in turning on this alternative promoter? And this is this study that I gave you out of Akimoto out of Zen Yan's lab. And so what they did here is really cool. They took this super incredible camera. There's a few $100,000 camera. And what it can do is it can actually look for light very sensitively. And it uses the light produced that we're going to see is produced by the luciferase activity. So this luciferase protein, which is a protein that's produced in fireflies. And the firefly protein is not going to be seen in a mouse except where you have put it in. And so what they did is they transfected the muscles on both legs with this construct that had this sequence attached to luciferase. Then what they did is they stimulated one leg, and then they took an image of it 0, 1, 2, 3, 4, or 24 hours after electrical stimulation. And what we can see is we can see the light that's being produced as a result of the stimulation. And what you can do is you can compare the stimulated leg to the non-stimulated leg, and that's now going to give you, this ratio gives you how much you've induced luciferase using this promoter. Okay, and what you can see is that after exercise, it begins to come up, and it becomes statistically significant after 1, 2, 3 hours after exercise. It starts coming down already by 4 hours after exercise, and by 24 hours, it's back down to 0. Okay, so this luciferase activity is showing us that the exercise has turned on PGC1 alpha activity because we're getting an increase in the transcription of PGC1 alpha using the PGC1 alpha alternative promoter here. Then what they did was really nice. They did mutations in 3 of these sites that they found, either the CRE, the cyclic AMP response element binding protein, or cyclic AMP response element, or they knocked out the 2 MEF2 sites within the promoter. They then redid the stimulation with the delta CRE or the delta MEF2, and you can see that either mutating the CRE or mutating the MEF2 completely prevents the increase in PGC1 alpha transcription. And so what that tells us is that in order to increase PGC1 alpha transcription, we need to increase MEF2 and we need to increase CRE and or. So we need to do one or the other of them. And so really now all we have to understand is we have to understand what these structures are and how they're regulated. And so we're going to start this by looking in the nucleus here, and you can see our gene coming in. That's the PGC1 alpha gene, and we've got this CRE, and we've got these 2 MEF2 sites, and the 2 MEF2 sites are going to bind the protein MEF2, as you'd expect, and the CRE can bind either this Krebs protein or ATF2. Either one of these 2 can bind to the CRE and activate transcription at this spot. We're going to talk about the E-box shortly, but really we're now talking about the 2 MEF2s and the CRE. All right, so how do we control transcription? The first thing was coming out of this Nature paper was looking at how CAM kinases can control basically MEF2. And so I'll show you that, because basically what they do is they take this HDAC5, which is a class 2 histone deacetylase. So it's a protein that deacetylates histones and keeps everything kind of in an inactive form. And if you don't have CAM kinase, you can see that the histone is located in the nucleus. When you add CAM kinase, now the histone is located everywhere except the nucleus. So now what you've done by adding CAM kinase is you've moved the histone out of the nucleus. If you mutate the histone in 2 spots, and these are phosphorylation spots where you can phosphorylate the HDAC, if this HDAC can't be phosphorylated anymore, now when you add CAM kinase, it stays in the nucleus. So what this tells us, these data tell us, is that phosphorylating the HDACs at these 2 sites, CAM kinase is going to drive the HDACs out of the nucleus. So that's the first thing. So CAM kinases can control these proteins called HDACs by phosphorylating them and moving them out of the nucleus. So now how does that affect either or MEF2? Well, here's the nice thing. So when we look at this and how it actually first, this is how this is regulated. So just like you'd expect, the CAM kinase isn't going to be able to move the HDACs out by itself. So the phosphorylation does something. And what the phosphorylation does is it basically labels the HDAC. And it tags it so that this protein, 1433, can grab it and move it out of the nucleus. Think of it as this. You're going into a nightclub. You are doing a little too much in the nightclub. You're now celebrating a little too much, and you are becoming a little bit obnoxious. And now the owner of the nightclub points you out and says, we need to get rid of that guy. And so what happens? This big old huge bouncer comes up. And, a big huge bouncer, that's like 1433. 1433 is the bouncer. And just like the bouncer who's, maybe Eastern European, no vowels in the name. So it's like this, no letters in the name here. All right? And so this big huge bouncer comes up and grabs onto you because you've got the mark on you because the boss put the mark on you. And now he grabs you, takes you out, and throws you out of the club. That's exactly what's happening here. Okay, so when CAM kinases phosphorylate the HDACs, what they do is 1433 then moves the HDACs out of the nucleus. All right, so that still hasn't told us what HDAC is doing and why moving it out of the nucleus is important. But this one is going to start doing that. Okay, so what happens here is here we go. We've got our CAM kinase without CAM kinase. HDAC5 is here. With CAM kinase, HDAC5 moves out. Then what they did is they did a sequence, a nuclear localization sequence that would always keep HDAC5 in the nucleus. So even when you have CAM kinase there, HDAC5 is in the nucleus. So what's going to happen with that one? And then they mutated the HDAC5 in such a way as that it didn't move out of the nucleus either. Now let's see what happens. Now what we're looking at here is this is the amount of HDAC associated with MEF2 or 1433. Without CAM kinase, MEF2 is bound to HDAC5. When you add CAM kinase, MEF2 is no longer bound to HDAC5. Now 1433 is bound to HDAC5. Okay? Now let's see what happens when we do this the other way. Now you put a nuclear localization signal on here, and even though the HDAC5 is still in the nucleus, it can't bind to MEF2 in the presence of CAM kinase. Again, that's the phosphorylation step. So the phosphorylation step causes HDAC5 to release MEF2 and to be picked up by 1433, but in this case, it's going to get shuttled right back into the nucleus. So in our bar example, this is like you getting thrown out of the club and then running in the back door immediately. You're right back in the club. The bouncer's going to go get you again, throw you out, but you're going to keep running back into the club. But you can't dance with that girl that you were dancing with because you're always, every time you get back into the club, just as when you're about to start dancing again, the bouncer picks you up, takes you out, throws you out of the club. Okay? All right. So the 644 is now one that has the ability to bind is modulated a little bit differently. You don't have to really understand what's going on here. The key one are these two. So when CAM kinase is present, it phosphorylates HDAC5 or the class 2 HDACs. That causes it to release MEF2, get picked up by 1433, and thrown out of the club. All right? And that's really what we're looking at is what's happening here. And so now this inhibitor, HDAC5, which is inhibiting MEF2, has been taken out of the club. Just like if we're thinking of our bar example, say it's you're dancing with this girl all the time, she doesn't like it, because she wants to dance with another guy. Now what's happening is the bouncer comes in, takes you, throws you out of the club, and now the other guy has a chance to dance with you. All right? And now MEF2 has a chance to do what it would like to do. And what it would like to do is it would like to increase the rate of transcription of specific genes, like PGC1 alpha. So that's how we regulate MEF2 using CAM kinase. CAM kinase phosphorylates HDAC5 that releases MEF2. Now MEF2 can do what it wants to do. It can go and change transcription. So how about this other thing, the CRE? Well, here you can see when you've got CAM kinase 2 here, and you look at this cyclic AMP response element binding protein, CREB, so CREBP, so this protein can bind to and activate that CRE. So when CAM kinase and CREB are together, boom, you've got phosphorylation. And that phosphorylation is helping CREB to bind to the CRE. Okay, similarly, ATF1, you can also have the ATF is also phosphorylated by CAM kinase. So you have two different things that can bind to the CRE. So when we activate CAM kinase, it phosphorylates CREB, moves it in, and it can bind to the CRE. It can also do that through ATF. And at the same time, I should say, CAM kinase is phosphorylating the HDACs. That's moving it away from MEF2. Now MEF2 can bind to the MEF2 sites, and it can increase transcription because now we have the CRE and the two MEF2 sites bound. So it looks like this. You do your muscle contractions, you get calcium released into the muscle. That calcium is going to bind to the CAM kinase. That CAM kinase is going to come in, and it's going to phosphorylate CREB, and the phosphorylated CREB is going to be able to move into the nucleus and bind to the CRE. So that's occupying the CRE. And calcium calmodulin through CAM kinase is going to phosphorylate HDACs, which are normally sequestering MEF2. The phosphorylated HDAC can no longer bind to MEF2. Now it's binding to this bouncer, the 1433, and that bouncer is taking it out of the club and throwing it out of the nucleus. When it does that, now MEF2 can bind to the MEF2 sites, CREB can bind to the CRE, and we get this alternative form of PGC1 alpha. All right? And so that's how calcium, through the calcium calmodulin kinase, is increasing MEF2 and CREB, or CRE binding, to drive PGC1 alpha expression.